In this video, we are going over the concept of proteins, which is the third biomolecule that we're going to learn about in biochemistry. Proteins in the body are polymers of 20 different amino acids because amino acids are going to be the basic units, in other words, the building blocks for proteins. Proteins have different characteristics and function, and even though proteins are built from the same 20 amino acids, the difference in what results in the different functions that we observe is the sequence itself of those amino acids. We know that proteins are structural components. They are involved in chemical reactions in the body. They even transport substances in the body. In order for us to understand proteins, we must first explore what are the functional groups present in the basic unit for an amino acid. When we look closely at an amino acid, the reason why they're called amino acids is because they have an amine group, they have a carboxylic acid group, and that's why they're amino acids. Those two functional groups are held by an alpha carbon, which is right here, and that alpha carbon has an alpha hydrogen. Each alpha carbon, in addition, has an R group. Here we see specifically the amino acid that has an R group that is a CH3. And that R group, which in other words is the side chain for an amino acid, is what makes each of them to be different. In the figure that we see on the left side of the slide, we can observe an image of all the amino acids that are present in the body. In addition, we have the full name, one-letter code, and three-letter code for them. These structures do not need to be memorized because a similar figure will be provided specifically in the exam. The important thing to note about amino acids is that those R groups can categorize the amino acids into different groups. So when we look closely at this image, we can see that amino acids can be categorized as nonpolar. And when we observe closely at the nonpolar side chains, which are highlighted in green, most of them are hydrocarbons. The only one that is not a hydrocarbon is going to be the one in methionine, which this side chain is called a thioether. Thio because we have a sulfur, ether because that sulfur is in between two carbon chains. Next, when we look at the polar side chain amino acids, we had a larger variety of specifically functional groups. We have some side chains that are alcohols. We have a side chain that is a thiol. And we have two side chains that are amides. Next, we have amino acids that are considered acidic. And the reason why they're considered acidic is because they have a carboxylic acid in the side chain. We can also see that there are side chains that are considered basic. And those basic side chains are going to have amines in their structure. Many amino acids, or I should say the majority of them, have a stereoisomers. And stereoisomers is something that we explored specifically in the review that we did for carbohydrates. Stereoisomers or spatial isomers are molecules that have the same chemical formula. The atoms are arranged in the same position, it's just that they are different in terms of space. The only amino acid that is not chiral is going to be glycine. Because when we look at the structure of glycine, glycine just have a CH2 at the alpha carbon, meaning that the side chain is a hydrogen. But the rest of the amino acids are chiral. They may contain one or more chiral centers. We can observe here the example of alanine, which is the amino acid that has the side chain that is a CH3. And we have the D enantiomer and the L enantiomer. Which this means is that when we look at this bond specifically, because the CNH3 is pointing specifically to the right, that's why this alanine molecule is D. If your NH3 is pointing to the left, then that is the L enantiomer. So the rules for determining D or L for amino acids is a little bit different from the one that we learned previously about carbohydrates. When it comes to proteins, there's a different structural levels. And the primary structure of a protein is simply the sequence of amino acids that are present in the protein. Each one of these amino acids, at its basic chemistry, what's going to happen is that they're held together by an amide bond. So there is an amidation reaction that occurs inside of a cell to take the carboxylic acid terminus of an amino acid connected to an amino terminus to then create an amide bond. And amide bonds are also known as peptide bonds and is what links two amino acids together. If you are given a particular sequence for an amino acid, it is important that you're able to define what are the amino acids that are present there. As you can see, a protein is going to be a molecule that has many peptide bonds. That's why it's also known as a polypeptide that has 50 or more amino acid and has biological activity. And as I just mentioned, the primary structure of a protein is the sequence of amino acids that hold together that the amino acids present. So here we have the example of specifically a condensed structure for a peptide of 4 amino acids. Understand that when it comes to amino acids, you need to know the pattern that exists in order to identify the amino acids that are present. Amino acids start at an NH3 group, and then after that, we have the alpha carbon, followed by an amide, followed by an alpha carbon, followed by an amide, followed by an alpha carbon, followed by an amide. Then we have an alpha carbon. If after that alpha carbon, we have a carboxylate, that means that your protein or peptide stopped. So when we're trying to identify the amino acids in the molecule, we just first identify the alpha carbon. And here we have 1, 2, 3, 4. And then from that alpha carbon, we must look at what is attached to it. It must be pointing up or pointing down. So we can see here that because we have a methyl group that is alanine, since we have a CH2, CH3 connected to another CH3, that is leucine. If we have a CH2SH, that is cysteine. Lastly, if we have a CH2, CH3, that is methionine. Make sure that you practice with the in-class worksheet that was given to you. In addition, we must explore what is the secondary structure, tertiary structure, and quaternary structure for a protein. Let's start with the secondary structure. The secondary structure of a protein has to do with the hydrogen bonding events that are going to happen at the peptide bonds. There are two conformations that can be prepared when it comes to that. The first one is an alpha helix. And an alpha helix looks like a winding staircase. This winding staircase is formed by the hydrogen bonds that are going to happen between the carbonyl oxygen atoms and the hydrogen atoms in the amide groups. And these are all in the peptide bond. On the other hand, beta pleated sheets are going to be structures that are formed also via the same type of attractive force. And what gives the difference between forming an alpha helix or a beta pleated sheet is going to be the sequence of amino acids that are present in the protein, and they may have propensities to form an alpha helix or a beta pleated sheet. The tertiary structure of a protein is going to be the interactions between the side chains of the amino acids. And these side chains of the amino acids can be involved in the following interactions. They include hydrophilic interactions with water. When we observe this, polar side chains can interact with water because since they share the same polarity of water, we can see that they can, via hydrophilic interactions, can interact with water. We tend to see the hydrophilic interactions happen in the surface of a protein. Hydrophobic interactions can also occur. Hydrophobic interactions are going to happen specifically when non-polar side chains are hiding away from water, and they tend to be in the interior of a protein. Disulfide bonds are specifically present in cysteine residues that when oxidized, they're going to create such structures. We can also have hydrogen bonding events occurring between polar side chains to each other in the protein. Next, we can have salt bridges. And when it comes to salt bridges, understand that this is the interaction between a cation and an anion that are present in the side chain of a particular amino acid within a protein. And this forms an ionic interaction, an electrostatic interaction, and that's what is known as a salt bridge. Notice that hydrogen bonds can also occur with the oxygen and the carbonyl of certain side chains, and also whenever you have side chains that are amines. Lastly, quaternary structures are actually present in molecules that have different subunits that have to come together in order to give the overall structure of the protein. This is not present in every single protein, but some require such interactions. In terms of shape, Proteins can be globular or fibrous. Fibrous proteins, which is what we see on the right, are going to be structures that are involved in structural components of the cell. And these proteins are known as fibrous just because they're fiber-like shapes. On the other hand, most proteins are actually globular, which means that they have spherical shapes. And because when we observe their function, which include carried out synthesis, transport of substances, metabolism inside of cells, that's why it is suspected that many proteins that are found in the human body are globular. Proteins can be classified based on their function, and the different functions that they have include structural functions because they provide structural components. Proteins can be contractile because they make muscles move. They can be involved in transport because they carry essential substances throughout the body. They can be involved in storage because they store nutrients. Some proteins can be hormones because they regulate the body metabolism. Some can be considered enzymes because they're going to catalyze biochemical reactions, and some are involved in protection because they recognize and destroy foreign substances. In table 16.1, you can see examples of each of the classes of proteins. Protein denaturation is a event in which proteins can have different treatments that results in protein unfolding. Protein denaturation can happen when you heat a solution that has a protein above 50 degrees Celsius. Understand that this affects specifically the secondary structure and the tertiary structure of a protein because of the bonds that are disrupted. Other treatments include acids and bases, and as we can see, because of the bonds that are disrupted, this affects the tertiary structure of a protein. When it comes to organic compounds, it can also affect the tertiary structure of a protein because of the bonds disrupted. Heavy metal ions also affect the tertiary structure. And lastly, agitation affects both the secondary structure and the tertiary structure of a protein. We can also see in the last column examples of each of the denaturation agents and how they can cause protein unfolding in a set of examples. Now for the last part of our conversation on proteins, we're going to focus in a specific class of proteins, which are called enzymes. Enzymes are going to be proteins that act as biological catalysts. So what this means is that many of the reactions that happen in the human body are going to have a fast rate due to the presence of enzymes in such reactions. When it comes to enzymes, let's understand that enzymes, when we look at them structurally, one of the most important parts of an enzyme is going to be what is called the active site, because in the active site is where the chemistry occurs. Since enzymes are biological catalysts, they are going to speed reactions, meaning increasing the rate by lowering the activation energy of the reaction. In the energy diagram that we have on the right side, we can see an example of how the catalysis of this reaction can be increased or the rate of the reaction can be increased just because of the presence of an enzyme. While the blue line tells you what is the activation energy in the reaction progress of the same reaction without catalysis, we can see how the lavender line, it represents the trajectory of the reaction in terms of its rate, by comparing the reaction process and the energy. We can see overall that the activation energy is lower because of the presence of an enzyme. When it comes to enzymes, they're going to catalyze a series of reactions. And just to briefly summarize the different classes of enzymes and the reactions that they catalyze, we can observe in the following figure how they can be classified. When an enzyme is an oxidoreductase, that means that it's going to do oxidation reduction reactions. When an enzyme is a transferase, is involved in the transferring of atoms. If an enzyme is a hydrolase, is involved in hydrolysis. If an enzyme is a lyase, it's going to add or remove atoms to form a double bond or a new ring. Enzymes that are considered isomerases are going to rearrange atoms, specifically make structural or constitutional isomers. And then lastly, ligases is going to use the energy molecule ATP in order to combine two small molecules. When we look more closely at the active site of an enzyme, let's understand that in biochemistry, a substrate is another word that we can use for reactant. And the substrate binds specifically at the active site via interactions. And we can see that what keeps the substrate in there is going to be the amino acids that are present in the active site. So we can observe that the interactions of the amino acids in the active site is what actually permits that the substrate is present within the active site in order for the chemistry to occur. There are two models for enzyme action. The first one is called the lock and key model. And the lock and key model is utilized by some enzymes in which their active site has a specific shape for the substrate that is going to bind. They bind to each other, making a substrate enzyme complex that works like a lock and key. And then the enzyme is able to produce the product and then is regenerated in order to continue the reaction. Other enzymes use an induced fit model in which instead of just being set like a lock, what happens is that they do have an active site that can hold the shape of the substrate. But in turn, when the substrate and the enzyme meet each other, they make an enzyme substrate complex that is more flexible. And the enzymes reshapes and hugs in a way the substrate in order to convert it into product. So out of the two different models, specifically the one with the induced fit model is more dynamic. There are different factors that affect enzyme activity. They include temperature, pH, and the presence of inhibitors. Let's understand that when it comes to temperature, enzymes are going to have an optimum temperature. For the enzymes in the body, it happens to be 37 degrees Celsius. In the case of pH, enzymes can work in a variety of pHs. So there is not one specific pH for all enzymes. What we are going to be focusing on as part of this chapter is the presence of inhibitors when we're looking at enzyme actions. In this course, you need to be familiar with 3 different types of inhibitors. We have to be familiar with competitive inhibitors, non-competitive inhibitors, and irreversible inhibitors. In table 16.12, you see a summary of what is a competitive, non-competitive versus irreversible inhibitor in terms of the shape of the inhibitor, binding to the enzyme, and the reversibility. Let's look at the diagrams that we have below. If this is the normal interaction between the substrate or reactant and the enzyme, competitive inhibition happens because your inhibitor is going to have a very similar shape to the substrate. So what that means is that it's going to be able to bind to the active site, and then the substrate itself cannot bind to the active site, and that's why it's called a competitive inhibitor. Competitive inhibitors are actually reversible, and how you reverse their action is by adding more substrate. In the case of non-competitive inhibitors, the shape of the inhibitor is not similar to the structure of the substrate, and then it binds away from the active site, but it affects its shape. And that's why then the regular substrate for the enzyme cannot bind. Now, when it comes to non-competitive inhibitors, they are not reversed by adding more substrate. You need to use chemical means in order to remove them. Let's understand that because they bind away from the active site, that is known as an allosteric site in the enzyme. So the binding of a non-competitive inhibitor is called an allosteric binding. And lastly, we have irreversible inhibitors. They don't have the same shape as a substrate. The important thing is that irreversible inhibitors, the reason why they are irreversible is because they form a covalent bond to the enzyme, and these changes are irreversible, they're permanent. To give you an example of one of the first irreversible inhibitors that was actually developed and used in treatment, that would be penicillin. Penicillin is an important antibiotic that when it was developed, it was affecting specifically bacteria in order to treat people that were having bacterial infections. But understand that throughout time, then penicillin, people started developing allergies, and other varieties or variants of the structure needed to be developed.